And I tell you my journey, why this whole thing actually started to exist, and why I think this is an interesting thing for us, but also for the FHNV in our area. So having too much data can actually be a problem. And there's a bit of a paradox, because as researchers, we all want to have more data, and we all want to work through more data. But then we find once we have this massive amount of data, we don't have the tools to do this properly. Or even at the get-go, some machine gives me a lot of data and I don't know how to deal with it. And I give you a short example. And then based on this, I suggest a generic approach how a research data infrastructure could look like. And then I think we get in more and more detail. So this, by the way, is Paris in the night. If I look properly, there's somewhere the Eiffel Tower, but I can't find it now. But the problem is that access to data, and I'm only looking for access to data. I'm not even talking about the data we're creating in our lab. Access to data is abundant. This is massive. We have easily about, what did I find? About 110 databases on Wikipedia alone, on biochemistry, pharmaceutical chemistry, toxicology, inorganic, organic chemistry, whatever you want to find. Here's an interesting thing. Let me start my pointer here. Ta-da. NCBI alone here. Where's my pointer? NCBI alone here gives you access to additional 50 databases I found on top. And some of them are like Interpro, which gives you an integrated database of predictive protein signatures, which you can use for classification and automatic annotation of proteins and genomes. Then we have OMIM here, which is a comprehensive compendium of human genes and genetic phenotypes. And R3 Data is like NCBI, also a repository which gives you access to another set of databases, which from time to time overlap, but they also are different. You can find the genome of the Neanderthal and compare it against the genome of modern humans, human beings on top. So there's nothing, if you can't find it, it doesn't exist. I think this is what it comes down to. So everything is there. And it's actually not difficult to find it, but to access it and use it is a different story. So I guess there's about probably 1,000 different databases available. And this does not even contain the different software pieces I found and software tools for various purposes, such building your own open source data website for this type of data, tools to annotate genes, biochemical pathway analysis tools, crystallography, gene sequencing. It's just unimaginable what you can find. That is the open access world. I haven't talked about the proprietary data access, which is mostly run by hospitals, clinicals, and analytic companies, and sometimes universities, which are closed-end databases, which you might or might not get access depending on the research topic you're looking at. And then we have commercial data sources. And we all know this, publications, super expensive, Scopus, Science Direct, Springer Nature, Science, Wiley Online Library, Elsevier, you name it. And we also have various databases like Gmelin, SciFinder, Beilstein, which are some sort of the bedrocks, at least when I was studying chemistry, the bedrocks of chemistry, where you could look up all sorts of information on chemical products and chemical molecules. So this is where we are. Now, coming back to the other side. So I create data. So we have enormous amounts of data, but the workflow of research today and industry, as well as in academia, does not really take care of this. And this leads to a really, really strange situation. So I had this example. So there was a researcher that was analyzing the up and down regulations of genes in certain cells. And the result was that there is several thousands of up and down regulated genes, which is not uncommon. But this is where the problem here started, because there was no tool that actually was able to draw together all the data into one set. So what happened was this poor person was actually forced to start a manual comparison of the genes to standard cells, and this already takes a long time. And this would have been not so bad, but there's some sort of a little wrinkle which makes it really bad. Because what I didn't know is that gene identifiers are not unique. This person as well didn't know this either. So before you can even start comparing genes to each other, you have to normalize them on a common name. And certain genes carry about 10 different identifiers, depending on who published something on it and then had the feeling they have to give this gene another name. So it could be the Christian gene, and it could be the Victor gene, it could be the Alexandra gene, and it's all the same gene. So there's another problem. So we have too much data, and in theory everything is available, but the sheer amount of data just collides with the current way we're doing our laboratory work. And I think that's a problem. I think everybody agrees that this is a problem. So the irony of this whole thing is that we live in a world where the amount of data is actually not going to be less. On the contrary, we will expect an ever-increasing surge of data through scientific progress in almost all areas. That's the whole idea, because we all think that with big data and machine learning and statistical and other means, we can actually have deeper and deeper insights into what nature is doing or not. But this will also mean that there has to be a convergence between how the research workflow works today and the data flow that is created. This is happening very slowly because if we go back to the digital journaling and laboratory journaling, we're still in a position where we don't have a proper digital journal. There are commercial products out there, but they do only so much. You cannot really integrate your analytics data. You cannot do a proper literature research out of this. So you can't do a lot of things, but some of them are really good at is just capturing what you do in the laboratory, but that's it. And are very restricted in which area you do this. So this leads to something also very unwieldy, which is what we call media breaks. You have the situation where you have your laboratory journal. Now you have done your analysis, and the analysis is done with mostly a very expensive piece of technology. And then you print out the result and glue it into your paper-based laboratory journal, and that's it. So you're going from fully digital electronic to back again to physical, and there's no way that you can activate additional data as you do. And this sounds all very wild, and I've done it, and I think everybody on the call has done it probably as well, who did some research. And that's something which must change, otherwise you're going to get drowned into data. And there's today different kind of strategies that lead to it. Some people just ignore the data. Some people try to work through it with Excel sheets, but there's no satisfying solution available. So let's look for a generic approach how we can do this. And I'm daring a bit of a look into the future. So we will clearly see there is more data coming our way. There's more data sources, and there's a higher granularity of data because the technology enables us to look really down, literally on the atomic level of the things we're looking at. Now, this development will have, and there's no might, it will have an impact on how we researchers are working going forward, both individually, but also in teams. Individually, in a sense, how can I record it? How can I keep it? But also in teams, we will see that also people from different backgrounds will more and more work together because the questions we're asking are requiring more and more different backgrounds. You need somebody who understands data better than you do, but you also need someone who understands analytics better than you do. And actually, it's nothing new for most of researchers. That's why we have this fantastic analytics laboratory from Götz, and that's where we have Frank Senna, who's I think also on the call, is doing this incredible, really cool stuff for the students. So this is already happening, but with more data coming. So impacts I see. We will see digitalization of lab notebooks, integrating experimental data, but also the spectra and other things, anything you can imagine. We also will see that in order to be successful, we need to pick up a certain level of data science skills, at least comparable to today's know-how on how we interpret NMR spectra or HPLC MS spectra or whatever other analytical tools you need. You have to have a basic understanding how they work. Otherwise, you can't perform anymore. And maybe there's some sort of a low-code environment thing happening, so that in case you cannot actually code programs on the computer, you can at least stick together different modules that give you the desired outcome. And that remains to be seen. So looking into my crystal ball, I could see a future that will look like this. Integration of experiment analysis and data. Shielding the researcher from the complexity of data access and data sources. So I'm coming back to my slide with several hundred, maybe 1,000 databases. How do you get access to that? This shouldn't be actually the problem with a research person. This should be the problem of a piece of software, giving you the ability to access those data without really thinking, how can I access the data? We need to lower there must be a lower barrier of access to machine learning and AI, either to perform repetitive tasks. So it's not only very useful for gaining new insight into a new set of molecules, but also helping you to perform repetitive tasks on and on with the same quality and not getting frustrated, not getting tired. And of course, what I call data wrangling, so working with data, making it clean, pre-process it also through technology. Because as we all know, the data we're getting usually needs some preparation in order to do this. Okay, I'm running. No, I'm still a bit of time. Okay, so here's my idea. We have a generic data research platform. We give access to relevant and high quality data sources. We manage access to our experimental data, and we support different users and different research projects. So all these items I see here would be some sort of the ingredients, high-level ingredients, how you can build a data research platform. And that would be even more important if you look at the different things we have to take into consideration. The project, what we're talking here, like Origami, is not supposed to experience a premature death. So it lives and breathes with your willingness to contribute. It must be a simple software architecture. The architecture must scale. And we have to understand, and that's very important, that the people we're inviting here, they are not all chemists, they're not all biochemists, they're also pharmacists and come from different scientific backgrounds, so we need to cater to them too. So the approach would be, we start with this open source project. We do a certain software architecture, and I was going ahead of myself at that point. But if you look at how I think this could look like, so we have an electronic laboratory journal, which is we're going to call Origami Lab, which contains its own database where you can have literature bookmarks, where you can store your experimental parameters, where you can draw data from other external databases, where you could do analysis, visualize, support for publications, do literature research, have a user management tool so that you can manage teams, and do all the other things that you can ever think of. And I left deliberately space here because we can add almost anything we can think of what we would want to do. Maybe a voice recognition system as well.